Welcome, everybody. I'm John Alpert, Chair of the Department of Psychiatry and Behavioral Sciences. It's a great pleasure today for us to welcome our very distinguished speaker, Professor Ron Davis from Stanford. The introductions will be made also by a distinguished speaker, Professor Scott Emmons. Some of you know Professor Emmons as Professor of Genetics and Neuroscience at Einstein, so a good colleague across campus. I wanted to mention that Dr. Davis, Professor Davis's talk is going to be broadcast to our basic science colleagues at Einstein, and he may indulge in some basic science, which is really the world that he's lived in and advanced for many years. It's also being broadcast to colleagues at Columbia, in addition to our clinical sites that will be watching. Some of you may know Dr. Davis's very moving story and the way that he's used his prodigious background in science to advance a very understudied, poorly understood, underfunded area that became of personal interest, unfortunately, to Professor Davis and his family in recent years. So we're very, very grateful for Professor Emmons' both introduction and also for coordinating Professor Davis's visit. Thank you very much.
Thanks, and thank you for your departmental involvement in co-hosting Ron's visit. I've known Ron since I was a graduate student in the early 1970s, and Ron was hired as an assistant professor in the department at that time. And probably you appreciate that much of what we know of modern molecular genetics, the techniques of starting with recombinant DNA, putting genes into bacteria, leading to everything we know of modern molecular biology, molecular genetics, molecular medicine, DNA sequencing, and so forth, originated in large part from the Stanford Biochemistry Department in the 1970s. And in that group, Ron Davis himself was a major contributor, and he's recognized as a major figure in development of molecular genetics in the last 30 years. His CV lists 508 papers, 55 patents. He just told me today that his Genome Technology Center has spawned 40 companies. His CV lists 23 awards, including the recipient of the Eli Lilly Award, United States Steel Award from the National Academy of Sciences. He was elected to the National Academy of Sciences in 1983, a recipient of the Genetic Society Lifetime Achievement Award, the Gruber Genetics Prize Award. His work has just been foundational in many, many areas, very innovative, and largely development of techniques and technology. Ron, 1972 paper was first paper to show that restriction enzymes create cohesive ends, which is how we join DNA together in recombinant DNA. A 1980 paper proposed that restriction enzyme polymorphisms could be used for human genome mapping. And that paper has received 12,806 citations as of yesterday. A 1995 paper described the use of cDNA for measuring gene expression. That paper has been cited 12,806 times. To give you a sense of the breadth of the contribution that Ron has made, I just glanced down his list of publications, and a couple of titles caught my eye. Here's a 1999 paper, Y Chromosome Evidence for Northward Migration of Modern Humans in Eastern Asia During the Last Ice Age. Okay. There's a 2012 paper, Aerobic Dive Limits of Seals with Mutant Myoglobin Using Combined Thermochemical and Physiological Data. Finally, there's 215 paper, Integrating Cell Phone Imaging with Magnetic Levitation for label-free blood analysis at the point of living. It's a remarkable breadth. You can see the influence, the importance, and how a person can contribute at an author level to 500 papers. It's such a widespread influence. Just to give you one more anecdote, I had a talk with Ron at the cocktail reception for a reunion at Stanford some years back, and Ron got something out of his pocket. He showed it to me, this tiny little metal object. He said, this is a centrifuge rotor. He was developing massive parallel DNA isolation, I believe, for his DNA sequencing projects. It just gives you an idea of the innovative work that has come from Ron. And so it's going to be very exciting to see how such an individual now approaches an intractable problem like chronic disease syndrome. So he gave a talk yesterday at Princeton, Immune and Hematologic Characteristics of Myalgic Encephalomyelitis, Chronic Fatigue Syndrome. And today's talk, it will be Progress and Challenges for a Myalgic Encephalomyelitis Chronic Fatigue Syndrome Diagnostic. Ron?
Well, thank you very much. Basically, I've been forced into studying this, and what I wanted to define is that this disease has a couple of names. Both of them are actually pretty bad. Technically, this is not correct, and this is a kind of a stupid name. It's just reflecting that they're fatigued, and a lot of people who say, oh yeah, I was fatigued once, they don't have chronic fatigue syndrome. If they said they were fatigued once and they're okay now, then they're incorrect. You almost never get over this disease once it starts. It starts frequently as in late teenage years, often from an infection. Mononucleosis is a common one. There's one big study that shows that 10% of those patients come down with chronic fatigue syndrome right after the infection. I had a graduate student in the lab that came down with mononucleosis. She was out for about 2 months. She came back to the lab, worked in the lab for a few weeks, and then came to me and said, I'm getting worse again. And what she thought was, well, she really wasn't over the Epstein-Barr virus infection. But I didn't see her again for 5 years. We did manage to get her PhD. It was difficult, but she was very smart and very dedicated, and she could do a lot from her bed. So this is the name used in Europe, and this is the name used in the United States. The problem is many people haven't heard of either of these, but to try to change the name is a problem, at least until we figure out what it is. So what a lot of people are now doing is using a combination name of ME-CFS to try to reflect both Europe and US. Now, it's not a rare disease, which I think a lot of people think about. It's actually a common disease. My estimate is probably around 1%. CDC says 0.7, but I don't like their statistics. I think it's underestimated. There was an IOM report for the Institute of Medicine, and it suggested from the data that about 80% of the patients are undiagnosed. It typically takes somewhere between 10 and 30 physicians to go to before you actually get a diagnosis. Many people give up. So the reason I am studying this disease is because my son has it, and he's a severe patient. So a lot of people say, oh, I've heard this is called yuppie flu, which means you're sick for a while if you're wealthy. That is not what this is. It's just that you often see the wealthy people because they can afford to go to a doctor, or 30 doctors or more. So he's very sensitive. This is typical. He's sensitive to sound. This was just opening his door and taking a picture. We were not allowed in at that moment. He has to cover his ears completely to block out all sound. He's touch sensitive, so it's really difficult to work with him. Smell sensitive, so we have seals on the door to prevent any smells from our house to go into his room. But unusually, he's not light sensitive, and that makes it a lot easier to take care of him, and I don't know why. Most patients are very light sensitive. They have to be in a dark room, and they often wear a black blindfold over their eyes in addition to being in a black room. These are the severe patients. They never go to hospitals. Most of them do not have medical care. It accounts for probably close to a third of the patients. And it's very common. So when we decided to study this, we realized we could actually get almost all these patients in our local neighborhood. We didn't have to go out and travel to different cities. This shows that he was drinking water at this time. He can no longer drink water. He can't eat. He has a J tube for feeding, and he has a Hickman port in his chest for IV fluid. We give him a liter of fluid every day, sometimes two. So most of these patients in this state stay this way for the rest of their life. Doctors say the good news is you don't die from this disease, and the bad news is that you don't die from this disease. Now that seems like a horrible thing to say. However, it reflects the mood of some of the patients. And that is, they know they're going to live a normal lifetime, but they're going to be in a miserable state. Suicide rates are extremely high. There has been work in England which suggested that it was a false illness belief. And what's interesting about this is that the best diagnostic criteria that works well for these patients is a test of post-exertional malaise. So in other words, if they exercise, if they do an exercise test, they're fatigued, they don't do very well. But if they come back after 2 days for that exercise test, they will do worse. So the hallmark of this disease is if you force yourself to exercise, you'll get worse. The treatment in much of Europe is exercise, and they get worse. And I just feel that that's a break of your Hippocratic oath. You should not do no harm. And it's still practiced, and it was only recently that we got the CDC to remove that recommendation from the website. This is an organic disease of some kind. It's very systemic. The immune system is involved, the brain is involved, the gut is involved, the muscles are involved. So to take this problem on, the problem is we don't know much about it. And I said, in order to really generate good hypotheses, we really need observations. And the scientific method is observation, hypothesis, and then a test of the hypothesis. So I wrote several grants to NIH to try to get funding, and they said the hypothesis was not very good. Well, no, of course not. It's not based on anything. It's guesswork. So we need observation, and I was not going to get it from NIH. And so I had to get involved in setting up a charity. So we got basically patients donating money. And so almost everything I'm going to show you is paid for by patients. And I really salute them because they've really stepped into the plate. And many of these patients are not wealthy by any stretch of the imagination. And they give donations of $5, sometimes $10. So we decided that maybe we should look at the severe patients because they've never been looked at before. And so, and then it's an expensive thing to do if we want to collect a lot of data. So we decided to look at 20 severe patients, hoping that that would be sufficient to collect enough observations that would be useful. For almost everything we do, there are lots of other studies with healthy people. We can use that. And we decided to use 10 just to make sure that that's basically not a control really to compare here to the severe patients, but to make sure that these controls look like controls that other people have done for other diseases. So it's really a test of our technology and procedures that we're doing things correctly. So we had to go to their homes. They are not coming to us. There's no clinics. And we went to each home. They were not far away, so that was useful because we then bring them back to the lab to analyze. When I put out the word that we're interested in this, we found several patients that were just blocks away from my own home. And I never knew it. And there was one severe patient about 4 blocks from our house, bedbound. Now, just to give a little bit of highlight for this, and comparing this to their quality of life compared to other diseases, and these use some standard scoring systems. Don't know how reliable they are, but they're the best we have at the moment, is really to, and these are just principal component analysis type things, and to show these are our controls. This is the general population, and here is the patients. And what I wanted to also point out is here's depression. So the patients are often diagnosed with depression, and that's probably totally incorrect. And it appears to be very far away. And when my son was still able to go to doctors, they would diagnose him with depression, and he said, you would be depressed too if your doctor told you were suffering from depression and you knew you weren't. It's very depressing. And that's what a lot of patients actually feel, that this diagnosis is not accurate. So in the case of the severe patients, we wanted to measure lots of things. What's also happening at the moment is also looking at families, because there are families with multiple affected. So that's a chance to see something in the environment in that patient family or something genetic. So there's something in common with these families that have 2 or more affected. That study is ongoing. And then I'm just not going to go into the details of these, but these are the tests that we ran. We're still adding to them, but these are the ones we picked out. So it's pretty extensive. And some of these tests are tests that we've developed ourselves, and they're not generally available. Oops, excuse me. So just show you a little bit about the behavior of these patients. It's easy to measure the cytokines. It's been done a lot in these patients. That's most of the publications are measuring cytokines. We use the Davis Immune Monitoring Core, which now measures 63 cytokines. And we looked at healthy controls, moderately affected, and severely affected. And the severely affected are the reds, which is on the outside of this. You don't need to really see the numbers. I know they're too small, but this is a visualization showing that they're actually worse off than moderate in levels of cytokines. So obviously these cytokines are causing a lot of the phenotype and maybe the fatigue. You can also look at all the metabolites, or most of the metabolites. This was done in a collaboration with Metabolon. This was the very first sample sent to Metabolon from my son. And what I expect every medical student and every biologist is to know this pathway by heart. This is the metabolic pathway. I know you can't see it, but you should know it. So what I've done is color-code it to give you a visual impression that it's all over the map, right? And so red is more than 2 standard deviations below healthy controls, and red is more than 2 standard deviations above. And you see it's all these pathways are being affected high or low. I'm just going to blow up one pathway that is probably significant, not just from this work, but from a lot of other people's work. I want to acknowledge that. And so this is the TCA cycle, and this is just one patient. And we do get some variation in this at different patients, but I color-coded this. You can see that a number of the components are low in the citric acid cycle. And that is frequently the case. If you look at a patient with a genetic defect in mitochondria, and we've done a lot of studies with a physician at Stanford on these, we do a lot of the sequencing, it looks just the opposite, that these intermediates are all very high, and that's because it can't process them. So there's something about this, it must be blocked even before it gets into the mitochondria to generate any of these compounds. But this could explain a fair amount of the fatigue. I don't know, I don't see any real evidence that this is the real cause of the problem, because we can't find a defect here, but it's probably a symptom. So I don't want to go, there's a huge list of metabolites that are all interesting, but it would take the rest of the hour to go through them. I don't think it's worthwhile. So I just pulled out a few things that I think give us a clue about the patients that may be interesting to other people. One of them is indole-propionate is quite low in the patients, some actually down here, basically undetected in the mass spectrometer. And when we first saw this result, being that all the patients have something low, there are a few healthy people that are a little bit low as well, but some are quite high in this metabolite. And so first thing we obviously do is, well, let's look up the gene. Well, that was an assumption that it was a human gene. The genome doesn't code for a gene to make this compound. It's coming from the microbiome. So about half of the metabolites come from the microbiome. This is produced by a Clostridium, and as far as we can tell, that Clostridium is missing in all of the patients. This compound is a neuroprotectant, and that may be significant. Because it's a Clostridium and sensitive to oxygen, most probiotics don't have it. You take quite a bit of special care to do that, because you've got to shield it from oxygen until you get it into the intestine. The other possibility, this is a small compound, it's made from tryptophan. It could, in fact, be supplied. And that might be the best potential treatment, is to supply this from the outside. There is a company now producing this. You can easily buy this compound, but it's not been tested in humans yet. Then there are two others. Lysine is very high, and hydroxyproline is very high. Now, that's interesting from the point of view, and this is just a correlation, that these are both involved in production of collagen. And especially this one is unusual in that it's present in, this is a common amino acid. And so this suggests that there's degradation of collagen. Consistent with that is an observation that there have been a few patients that appear to be like, symptoms-wise, like all the others, but it's not clear at all, if I'm talking with the physicians, that this is true for everybody. In fact, they think it's only true for a minor fraction of the patients. And that is that the collagen connections in the neck are insufficient to hold the head up. And the head falls down until it impacts the spinal column, and it impacts the spinal column in the brainstem. And so this was discovered in one patient who had a thyroid tumor. They removed the thyroid tumor, and in recovery, if she turned her head, she would black out and stop breathing. So that was now a crisis. They had to figure out what was wrong. They did a lot of studies on her and realized that the spinal column was impacting the brainstem. So they simply put metal rods to lift her head, and when they did that, all of her symptoms went away. So that's also an indication that this is an organic problem, not something as a false illness belief. Now, it's very clear that there's a lot of things going on in the brain, so there's a tremendous opportunity, I think, for neurologists and people who study the brain to help in this. Brain's complicated. I certainly asked for volunteers for doing some biopsies. I found none. So it's really hard to study, and we don't have model systems. And I'm not sure that we should even try, because this may be very much a human disease, and we may be misled by looking at other animal models. Possibly we can do some stuff with cell culture. So the plan of how to operate, this is just some preliminary data. We have a lot more. I'm not going to show that. What we have been doing is taking all the data that we get and posting it, and before publication. So we have a website that you can look for all of the data, and that anybody in the world that does scientific research can access that data. Here's the plan of what we would like to do. We first like to focus on finding a biomarker, something biological that you can test for that would tell you that you have the disease, or at least you're not healthy. Something is wrong with you. And then focus on all these ideas about treatment, like maybe the indole-propionate. We should work with a company to develop that to see if that would help the patients. And if we can understand the disease sufficiently, then focus on trying to do a cure. And even if we can't do a cure, we should also focus on prevention. If you can prevent it, you don't need to cure it. But that's not going to help any of the existing patients. So this is sort of the order that we are thinking about in terms of what we should focus on it, and we haven't done anything with prevention yet until we try to get some headway on these. So in terms of the biomarker assay, we really need one because the problem is when these patients are able to and go to a physician, they run a lot of tests. And that's a test they will run on many other patients on an annual basis. And when you do that test, they look healthy. So if you run those tests, and we have many times on my son who's bedbound, the doctor who just looks at the test and not visiting the patient would say, oh, this is a young man in absolute perfect health, low cholesterol, low blood pressure, all the markers are absolutely in the mid-range. So there's nothing wrong with him, except for the fact that he can't eat and he can't drink and he can't move, and he's bedbound for years. There's a problem. So that was our first idea, is to find a way to say that these are not healthy. And it's kind of ironic that those tests are about the only tests that come out normal. Almost everything else we run is abnormal. But mostly the focus is to distinguish them from healthy. And then at some point we need to try to distinguish them from other diseases. That's a little harder to do than a lot of other diseases, but that's something we will try to do. While we were working on the genome project, we developed a technology that can be used for a number of different things, and we have used it, and it's called a nanoneedle. And it was done several years ago, where it's made by nanofabrication, how you make computer chips. It's a little tiny chip, and it's made by layers of silicon oxide, an insulator, and then 2 gold electrodes. And these gold electrodes are about 50 nanometers in size. So you really can't see these, they're too small. And then we make these multiple of these electrodes coming into a trough, and we put blood in this trough and measure electrical impedance from the blood. Unlike a lot of medical tests which do one measurement, it's an endpoint measurement in many medical tests, those kinds of tests can have 10% false positive and a 10% false negative, not uncommon. And they're still used. But in this situation, what we do is that we have 2,500 of these electrodes independently being measured. So those are all independent measurements. And we take those measurements 200 times a second. So we have millions of measurements, and that helps us to not get false positive or false negatives. So when we do this run, and it's done with taking a blood sample, we remove the red cells because we actually know that the red cells will give us a signal, but in the opposite direction. So we either run this on red cells or white cells. We've chosen to run it on white cells because we know the immune system is involved. So we all we do is eliminate the red cells and put the plasma and white cells in a trough. And then we add sodium chloride. The idea behind this experiment was the patients are easily fatigued. Maybe that's true for all of the cells in the body. It's true for maybe the brain, it's true for maybe the immune cells. So if we add sodium chloride, it will get into cells. Sodium is a bit toxic to cells. They have to pump it out. And they will pump out the sodium and pump in potassium. In order to do that, pumping requires ATP. And so that will stress them because they have to use a lot of their ATP to keep the sodium out. And in principle, we do a titration of sodium, and then in fact, the first test worked. So we haven't actually optimized it, but this is what you get. So the blue is the healthy patient's blood sample. It basically doesn't change at all. There is a change as soon as you add the sodium, and that's just because the ionic strength of the media changes. And then after that, it's pretty flat. However, the patients start out the same, and then at different times, and we don't know why the timing, they will increase in electrical impedance. Electrical impedance is used because it's very sensitive to a lot of different dynamics of a cell. If the capacitance of the cell changes, you'll see impedance change. If the resistance changes, you'll see an impedance change. If the lipid content changes, it will change. If the size of the cell gets bigger or less or smaller, it will change the impedance. So it's a very sensitive measure of any kind of perturbation without having to know what it is. And all 20 of our patients show this. We have gone back now and done as a validation study 20 other patients that have the disease and 20 other healthy controls, and we get the same result. They all show it, and none of the healthy controls show it. What we don't know yet is do other diseases show this behavior. One of our problems is that this ME/CFS is caused by a stressor, an infection, an auto accident, surgery, childbirth. There's a variety of stressors that seem to be able to trigger this disease. Having another disease like MS is definitely a stressor, or cancer is a stressor. Can they also induce the same state? Generally speaking, that will be ignored because if the patients are fatigued, they're fatigued because they have cancer, or they're fatigued because they have an autoimmune disease. But it's possible that that initiates a different physiological state as well. So it makes it difficult to actually interpret the results. Then the question is this really, the assumption we made initially that this was in the cell, but it's cell with plasma. So which is the important component? So we did a plasma swap experiment by taking ME/CFS cells and putting them in healthy plasma and vice versa. And it tracks with the plasma, not the cell. And you can see that the best one is just here. When you and this is the classic this is the classic response to the ME/CFS cells. But if you take those cells and put them in healthy plasma, it's pretty flat. So now it's not absolutely completely flat, but that's because we just took them out of the plasma, so they have been exposed to it. But it seems that there's something in the plasma. And that's particularly interesting because it means we probably can identify by basically biochemistry of what it is. We haven't pursued that yet because of our instrumentation isn't sophisticated enough. We're only able to do 2 samples at the same time. The electronic instrument's commercial, it costs $30,000. It's about almost the size of this podium. And you can't do biochemistry fractionation with only 2 samples. And so we have to redesign the electronics, and that's being done at the moment. That's just an engineering problem. We have a prototype already built. It will cost us about $200 for this prototype to build it, and it can do 100 samples simultaneously. So that will be sufficient to do fractionations. Now we've looked elsewhere for changes, and I'm not going to go through them all. I'm just going to go through a couple here. And one of them is looking at red cell deformability. One reason for doing this is that there are a number of diseases that show red cell deformability characteristics. And one of them is sepsis, and looking through a lot of the data, there's a lot of similarities with sepsis. It isn't sepsis, but it looks a lot of similar events. And of course, septic patients are fatigued. So we decided to develop and look for a deformability. To do that, we really put together a team of people to help us do that. Eric is in chemical engineering, and he designs jet aircraft wings. which means he deals with airflow over surfaces. Deformability is going to be blood going over surfaces. So he can do a lot of the modeling of how we should design the system. He also has access to the Defense Department's computer systems. And so this is a very intensive computational problem. And also some of these other individuals are experts in biochemistry or blood and engineering. And Juan Santiago is a mechanical engineer and is really good at designing instruments. So this was a good team that we put together to look at it. The first thing was to look at the theory about the problem is in a capillary, you go from a large artery down to a capillary, and does it depend on where the blood cell is before it enters? And that will cause us variation, and we can't deal with that. So this was modeled, and that's what this shows you, the modeling. And what we're looking for is how far do we have to go before it doesn't matter where it starts, it will in fact be the same. And that helps us in the design. When these red cells enter, they go through a capillary that's smaller than they are, and so they have to get squished. It's interesting to understand why that is. I don't see why evolution made it that way, so it seems as though there's some physiological reason why you need to squish them. But when they start going through the capillary, they look like they kind of develop a bullet shape as they move through. And and then we also looked at modeling how far do we have to make the capillary before we can actually detect it. So we've designed a number of things. These are just a few of them just to give you a physical feel. Here's the tube, and then we go down to the capillary, which is right here. These experiments using a 5-micron wide capillary. Sometimes it's 7 microns, but the red cell is much bigger than that. And then we decided to try maybe multiple ones. I don't think we're going to use this one. It's a little too hard and complicated to interpret. So we'll use something of this type. The very first experiments were just looking at the red cells in buffer, but then we switched to looking at them in plasma. And in that experiment then shows a significant difference in deformability. The first experiments that Juan Santiago did with a much better instrument that can measure a lot more cells, those were done in buffer, and he doesn't see much difference. So we now think that there's something from the plasma that's contributing to the deformability, and if you take them out of the plasma, they become normal. So again, we have a situation where we can look for what is it in the plasma that's causing this effect. And that should give us some understanding of the pathway involved in this disease. Where did that compound or protein come from, and how was it made, and so forth. It's all about tracking back. Find a problem. Whether this is very significant for the patients or not is a little bit irrelevant at this moment, is that then you see something that's very different, very consistent, track it back. And that's sort of you do this genetically or you do it biochemically. There's another thing that might work, and that is, and I can just show you this. This is a commercial instrument, and what it is basically a little microscope, inexpensive microscope, that has a good working distance, and you look at the nail bed, and this is used for physicians to diagnose autoimmune disease. And you have oil on the nail bed, and you put the finger under, and you try to get them to hold still. And if you don't hold still, then we can actually stabilize the image. You can see capillaries under the nail bed. And if you look closely, you can begin to see the red cells going through the capillary. And at first pass, what you see is that healthy patients, the video is not working in this one, are much faster than the patients. So we have to look at that and all the different parameters of why that may be. But it might turn out to be a diagnostic tool, a very simple one and a very cheap one. So these are just two examples of things that we found. We have found a number of other things that work, and I don't want to go through all the details of all of these other ones. Just to say we have a whole bunch of tests that we run. We have some new ones now we're going to try as well, and what we need to do is compare them, and we call it a bake-off. Which one works the best? And what this offers you is a possibility that if you find some deviation that there are patients where it doesn't work, maybe another test does. And so you can combine tests to make it more accurate. And we've done a lot of commercially developed tests, and always what you shoot for is very high accuracy at the beginning. And so, and we just in the process of doing this. One of the interesting one is lactate. You frequently find lactate at higher levels than in healthy people, and in blood or in sweat. And so that's a very simple thing to measure. So what we plan to do next is really, we've already done the validation study, and that works. And now we're starting to look at screening patients with other diseases, and we haven't yet started that. That's been delayed because we have to fabricate more of these chips, and they're fabricated by a graduate student, and the problem is when the graduate student leaves, you got to get a new graduate student, and then you got to train them, and that takes almost a year. But that we now have a new batch of chips, and we can restart that. So these are the things that we're currently doing with this, and we'll get back on track, I think, right away, because we now have. And what we will start doing, and we have started that with a small number of things, is to screen FDA-approved drugs. And how we plan to do that is there are a few treatments out there. They don't necessarily work very well, but it would be of interest to look at what before and after treatment with those drugs with our various patterns, and to see if we can actually see an effect. The other thing, which I think is more likely to be productive, is to do this in vitro by putting the drug into, and most of these things are blood or other kinds of body fluids that we see an alteration in, and see if we can switch that alteration. Blood is the biggest one. Because we see a number of effects in blood. And that would allow us to do all of the FDA-approved drugs. And I want to focus on FDA-approved drugs because we probably could use them off-label with the patients. If we find a pathway that's defective and we need a new drug, that's by all means do that, but typically it will take something like 20 years. It'll take a year to come up with a new drug, and then another 19 to get it approved, and probably at least $1 billion to do that. So I don't object to people trying a new drug. It would be incredibly useful, but we need to find something faster. So that's the reason to focus on the FDA-approved drugs. We also are going to look at natural products. These are basically extracts from plants. If we have a good biochemistry assay, I don't care that it's a mess. Does it help? So we're making connections with China and India because they have a lot of these extracts as well as anybody else that has extracts. And likely we would be able to use these if we can find something that works. Now let me go to some of the FDA screening that's been done. Because we have found some things. And one of them is Copaxone. Copaxone is to treat MS. It's a random polymer of these 4 amino acids. This is presumably the amino acids in the binding site for the antibody. And the idea behind this is if we make a polymer of these same amino acids, then there will be a decoy, to the antibody and prevent the antibody from reacting with the body. That was the idea. And if you make a random polymer, it's a lot cheaper and simpler. So, and it's used, and it does help. The only problem is that it has to be done, it's a long polymer, it has to be, you can't take it in the stomach, it's digested, so you have to inject it, or give it IV. And when we put this, this came from suggestion from Bob Navia, who's a mitochondrial and metabolomics individual in UC San Diego, because he had a person working with him that was diagnosed with MS who was taking this drug. She got a lot better on the drug. And then she discovered that she does not have MS. She has ME/CFS instead. So why did this work? And unfortunately, she became allergic to it because it is a protein. So she had to stop taking it, and all her symptoms came back. So she had some of this left over. She brought it up to us because she thought it was very interesting, and so that was our source of it at the beginning. We've since actually synthesized this. It looks exactly like the commercial. And then when we put it in our assay, here's the patient sample. If we use a low dose of this just to see what would happen, it shifts it in time, but the height is about the same. And if we put in the concentration that's used in the patient, it flattens out considerably. So that suggests that there is an effect on this drug. We've tried lots of others with no effect at all, so I'm not going to show you the negative controls. There's a lot of compounds that don't affect these pathways. Bob Navio also suggested that maybe we try Cerumen. The drug developed in 1906. It's certainly off patent. It's made by Bayer. It's used to treat African sleeping sickness. And of course, one of the interesting things, if you look at the symptoms for African sleeping sickness, they look identical to ME/CFS. One of the hallmarks of ME/CFS is the patients switch light-dark cycle. They're up all night and sleep in the daytime. That's why this is called sleeping sickness as well. However, this is caused by a trypanosome, and it will kill the patient. So this isn't African sleeping sickness, but it could be something related. And that does also have some big perturbation in this pathway, much more than than many other things we tried which don't do anything. So these are two candidates to be explored further. We can't yet get Cerumen. Bayer has torn down their manufacturing plant. They're making a new one. There's a new plant in the US being made, but they're still in the early stage trials with their Cerumen production. There's not much side effects with Cerumen used at these concentrations. It was tried as an anti-cancer drug, I think at almost 100 times this concentration, and the side effects were severe. So a lot of people say you don't want to try it, but they're looking at the symptoms. Oh, no, I'm sorry, cancer was HIV, excuse me. They don't want to try it because they see the symptoms, but those are a very high concentration. Not surprising, there might be a lot of symptoms. Another compound is called SS31. It's in clinical trials currently for congestive heart failure. It supposedly repairs the mitochondrial membrane. And I showed you earlier, there's some indications that the mitochondria may be involved. And so and I'm pointing out that it's a tetrapeptide, and it has tyrosine and lysine, just like Copaxone. So I don't know if there's a connection there or not, but it's something that we'll explore. These are both, Copaxone and SS31 are easy to make in the laboratory. It has a dimethyl tyrosine to be a better free radical scavenger. So it repairs the membrane and also decreases oxidative damage. The code hasn't been broken yet in the congestive heart failure, but a lot of the patients in that trial are getting much better. If you give it to patients with mitochondrial defects, where their mitochondria are very disorganized and their membranes are very disorganized, they become much more organized. They look like young, healthy mitochondria. So that's when they discovered that it was repairing the mitochondrial membrane. If you give this IV, within 5 minutes, it's gone from the blood. So it's absorbed very rapidly into cells. And then if we look at the results for that, this is the concentration used in the trial, congestive heart failure trial. Here's again the path that we see for the patient, and now that makes the sample completely normal. What it will do to the patients is the next thing to try to figure out. It doesn't have any side effects when used at this concentration, so it looks like a totally safe drug, even if you have to continue to use it for life. That we made other detectors that we're exploring, mainly for patients to have as wearables to make sure they don't overexert themselves. I won't go into much detail of this. This is looking at sweat. And so we can make a sweat sensor to put on the arm. This is a large flexible circuit board for our first pilot. This was made in collaboration with UC Berkeley. We had a lot of undergraduates do a lot of testing, and we had them go out and run, and we taped the circuit board to their forehead. It was interesting that at Berkeley, nobody noticed that these people were wearing circuit boards on their forehead. And that's UC Berkeley. We don't have to have it this big. This is just a test run. We can make a very, very tiny device with a wireless connection. And that can be directly transmitted to your phone, and you can look to see where you are. There's a lot of things we probably can set it up to measure. These are very simple, I think things to do. We've also looked at hair. The hair is really easy to do because you can get it from any place in the world. You simply have people cut a gram of hair off. You want it next to the scalp, so that will tell you what's happening recently. And you put it on an envelope and mail it. And when we get it, we open the envelope, put it in another container, and send it to a company for mass spectrometry. So it's a very simple and it's very inexpensive. We also, for the local patients, will take a blood sample as well as the hair. Some people don't like hair, and that's because hair can get contaminated. But we're not necessarily, so if we see something high, we have to be a little worried about it as external contamination. That's why you also use the blood. And this is just one result from that beginning study, because it was so prevalent. We saw it very quickly, and that is a lot of the patients had high levels. This is a WHO safe level of mercury. They weren't super high, but they were above that safe level of mercury. And so the question is, why do they have mercury? And so we contacted a number of the local patients that we saw, and we found that when we see high mercury, they also eat a lot of fish. And they eat a lot of fish because they think fish is easier to digest, and they think it's healthy because it has omega-3s in it. So they try to eat a lot of fish, and now they have mercury. Now, the mercury is a toxin, but the other thing we noticed in this experiment, because we're doing all of the elements, is that when the mercury is high, the selenium is low. And we do know that mercury will bind selenium. So that might be good, that the selenium is binding the mercury because it tends to inactivate the mercury. But we are worried about the low selenium. There's one case here where we see high uranium and a few patients. And here's one from Finland that's quite high. We have since found a patient here in the United States with even a much, much higher uranium level than this. It appears that it's probably coming from well water. So this is natural occurring uranium. The problem with that is that it's an alpha emitter. And if it gets incorporated into the body and it still emits alpha particles, it's very carcinogenic. And so we have found one person with high uranium that was just diagnosed with cancer. So it's not a good thing to have. And it's easy and cheap to test your well water if you're using that source. But apparently uranium can be a contaminant. And one of them was in California, and one was in Massachusetts. So I'm not sure there's a location you can easily be safe at. It just depends on where the water from the well is coming from. But the problem with selenium is that there are a number of essential enzymes that use selenium. And one of them is the conversion of T4 to T3. And a lot of the patients are low in T3, and some of them actually improve if you give them T3, and that is a selenium enzyme. And then there's a number of antioxidants that are selenium enzymes, in addition to a transporter, which is a selenium enzyme. So low selenium could cause oxidation and low T3, and that is a common problem in the patient. So we don't know that the low selenium is a cause for or contributor to this. I'm sure it's not, these are not that low, but it might be contributing to the problems. And it's easily and cheaply corrected. You just have to know. And you can't just go take selenium because you might be low, because selenium at high levels is also a toxin. So you need to put it at the right physiological levels, which means it needs to be done by testing. Lastly, I'd like to turn to thoughts on trying to cure this disease. And what a lot of physicians would try to discourage me is saying, well, you don't cure diseases. Well, you don't cure chronic diseases, but part of the problem there is that you don't know what's causing it. And I think it's probably, except for random chance, you're not going to cure a disease unless you know what's causing it and target something. So there are cases, certainly with infection, we can cure. We know what organism is infecting, we give a drug that kills the organism and the patient's cured. And that's been going on for a long time. Even genetics, there are a number of cases where we know it's genetic defect, and if it's in a pathway or something, a metabolite, we can intervene and give that metabolite either one that's missing that needs to be there or one that's toxic, and try to reduce the amount of the substrate for that toxin, like PKU. So there are a number of genetic diseases that we can actually effectively cure by just intervening. And we're going to get much better at this as we learn how to do genetic manipulation, and that's coming right around the corner. So to try to get more into what might be a cause, of course, is to focus in on the infection and the genetics, because if it's one of those, we have a chance of curing it. So we don't want to miss that. And so one of them, we can do a lot of this by simple sequencing. We have in the 20 patients, we've sequenced their entire genome, excuse me. That was done by Illumina for free because they know what we're trying to do. And they did high quality sequence and gave it to us for free. But now we look at that data and find things that are suggestive of problems in addition to the possibility of infections. So the problem is that it could be something that nobody's ever seen before. So we've got to be open-minded to that. So we have to have a very sophisticated, very sensitive, and a very inexpensive test, because we're going to have to look for a lot of different things. So the way we do this is that we do this with what may be called multiplexing. So instead of doing one sample at a time and one organism at a time, we set up a system where we can do a lot of things simultaneously in one tube at one expense. And so the target we have here is to be able to do 1,000 regions, which means 1,000 different regions from different pathogens. We often do 10 regions on each pathogen to make sure we don't miss it. And so we can do 100 pathogens simultaneously. And we set it up to be able to do that with a nanogram of DNA, so it does not take much sample. And also our cost is set at $10. Now, the reason we put these parameters here is that if you do newborn screening, there's a blood spot on a piece of paper, and you can test things by punching out a small punch from that blood spot. And that punch will have a nanogram of the newborn's DNA. So there's where you're set the limit at that. And then the other limitation is that, at least in the state of California, you have to do any of your genetic testing for $10 because it's going to be paid for by the state. And so what we would like to do is to test many of the childhood diseases and metabolic. And those are already being done, but they're not complete. So what they often do is look for common polymorphisms that cause defects in the gene, but they miss the rare ones. So what we should switch it to is sequencing the entire gene, and so we don't miss any of the new mutations that could be there. And we would like to do it on all of the different diseases, and we'd like to be able to do that on everything from one punch and 1 nanogram of DNA. We've already set up cystic fibrosis, where we can sequence the entire gene. That has been used at Stanford for all newborns for quite a while, just as a test case to see how well it works. It works pretty well. It's now being looked at by the state of California. So the way this is done, and this is for the DNA jockeys that may be watching, and they can appreciate the complexities of somehow to do these things, but why you can make them cheaper. So normally you do a PCR and you have a primer that goes to the area you want to amplify, and this region is usually the length of region you can sequence accurately by any of the machines. But we want to multiplex it. So if you put this in at high concentration, then you get primer dimers, and that fouls everything up. So you have to put this in at very low concentration, but then it's hard to amplify. So what you then do is the primer has a tail on it, and this tail is a this is a homopolymer tail. And all of the primers have that same tail. So a homoprimer will hybridize very quickly. And now if you amplify with these fragments and then you switch it to the homoprimer, you can then amplify up the sequence, and you can amplify anything else you put in the mixture. So now the advantage of this is the fact that when you do with a homoprimer is that the sequence on this strand is homologous to the sequence on that strand, and it will hybridize. And when it hybridizes, it will take it out of the solution and will not amplify. So when you get a primer dimer, they snap back instantaneously. And the rate at which it hybridizes is dependent upon its length. And that's all been worked out physicochemically. So we know, depending on how long we make them, that determines how long we have the cycling. And so now you can cycle it so that if you have something in between, it works, and if you don't, it goes out of the solution. So that gets rid of one of the major problems. And then you can take this and go right into the sequencers, and this is a case of the Illumina sequencer. We can then put on the sequences that need to be on in the Illumina sequencing. So it makes a single tube reaction, and you can do 1,000 regions simultaneously. And so right now that cost is still $10. So we've used that a lot, but we've used other techniques that I won't go into but we've searched a lot for new pathogens just by bare sequencing. We haven't found anything. We've used that with the blood center up in UC San Francisco. We've done all the DNA viruses. We haven't found anything. I mean, yes, of course, everybody has the herpes viruses, but the patients have low levels of that DNA in their blood than healthy controls. When we look for new viruses, we find the TT viruses. That's a good, healthy internal control because everybody has TT viruses. If anything, the patients have lower levels of TT virus than healthy control. We've done a lot of parasites. We have not done getting probes for those. We've done all the trypanosomes. We're still finding other parasites, and in fact, we found some that have not been sequenced yet, so we're going to sequence them. And so far, we haven't found any parasites. Ultimately, what we may be able to do is to put all of these together into one reaction, and that would test for all pathogens in one test for $10. Now, this tests for DNA, and so there are cases that may have the DNA can't get into the blood, but the blood is actually the sewer of the body, so anything that gets destroyed or inactivated will wind up in the blood. So there are companies that are now doing this and claim that every infection, no matter where it is, there will be DNA in the blood. Not the organism, but you kill the organism and the DNA is very stable and you'll find it in the blood. And you'll find the RNA as well if it's an RNA. So we're doing the RNA viruses. We've just about got that ready to go. The funguses need to be done, and so do bacteria, and that's a little harder to do because there's so many of them, and we have to figure out what we're going to do to look for those. But we're continuing to do that because if it's an infection, we could cure it, I think, or at least we have a good target. The other thing about genetics, we looked at through all the genes, looking for something that might be relevant. And especially that something that is defective in all 20 severe patients. And we have found the first one, which we thought was significant, maybe significant, is that all the patients have an IDO2 mutation. So that's a gene that's involved in tryptophan to guanine. And that was significant, but not significant enough. So we did 46 additional ME/CFS patients, and they all have a defective gene. So the problem is, of course, that it's common in the population. So, but this is actually statistically significant. We want to add to this and do more patients. Of course, the problem will be that if we get a misdiagnosis, that may not have it. So we have to be careful about how we interpret this. But this is a first pass thinking about how could an IDO2 mutation cause this disease. So if you look at the pathway biochemically, here's what it does. Tryptophan is converted ultimately to kynurenine, and also tryptophan is converted to serotonin. So this pathway is maybe very important for components that might be in this disease. Kynurenine regulates the immune system. So at low kynurenine, you can't suppress autoimmunity. It's also involved in the brain. At least in mice, if you add kynurenine to the brain, it can suppress inflammation in the brain of mice. Hasn't been done in humans. There has been a clinical trial of kynurenine injected into the spinal fluid, and it does appear to decrease migraine headaches. That has not been published yet. So this is a pathway that has components in it that needs to be further explored. It's not the only one. This is a little strange because there are two enzymes that do the same thing. So because of that, the geneticists have ignored this because they say this has too many mutations in it to be biologically significant. And so if you do automated analysis of your sequence, it doesn't even show up because it's excluded. And the biochemists exclude it because these enzymes do the same thing, so it can't possibly be involved. And you just always got to be really careful with those quick-minded conclusions. So if you go back and look at the biochemistry of this, which an awful lot of people do not do, there's a paper published in 1967 that shows that IDO1 is substrate inhibited. That's really odd. So as you increase the tryptophan concentration, you increase the throughput in the pathway until you get a critical point, and then it gets inhibited. However, you have IDO2 in the same cell, and it is not substrate inhibited. But it works at much higher tryptophan concentrations. So the net result in this cell is the green line, and it looks just like a normal enzyme curve. So if you just opened up cells and measured it, it would look absolutely okay. But many people have a defect in IDO2. Those people look different. Those people look like the classic pure IDO1 curve, where at high tryptophan, you don't make very much kynurenine. And so that will act like a mutation in IDO1, and kynurenine is also used to make NAD. So this could be a way to effectively generate what looks like a mutant phenotype. Now, this was all put together by Robert Fair, who does nonlinear system theory. He's a consultant for this. He consults for pharmaceutical companies. So he can do a lot of modeling. And one of the questions we ask, well, if you're down here somewhere, what are the chances you can get back over here? It's very low. And he models it that there's a chance that you could get over here just by random processes, but it would probably take 100 years. So that's not an effective treatment. So basically, there are just these key points, is that the severe patients have, on average, 1.7 broken copies of IDO2. There are many more patients that have defects in this gene. So effectively, the IDO2 acts as a backup if you ever get into high tryptophan. Now, what also is interesting about this is that when you get a viral infection, which causes this disease, it affects the tryptophan pathway. It will either lower or raise tryptophan. So this whole pathway is affected by infection. So that's another connection. Another connection is that you can inhibit the IDO1 by a drug. And it was this pathway of IDO1 is activated in cancer. And it's thought that it's activated in cancer because it helps them to escape the immune system. And so the thought was, if I inhibit IDO1, then they won't escape the immune system and you can help cure cancer. So they made a drug that was very effective at inhibiting IDO1. When they used that on patients, it caused extreme fatigue. So that's, again, another correlation, not causation, that this pathway might be involved. But in any event, this is an example of something we call a biostability. And it means that you can get a healthy state and a disease state, and there may not be easy switches between the two. And if you have this, if you're locked into this other state and you can't get back to the healthy state, then you have a chronic disease. So this just raises the issue that maybe these pathways that are substrate inhibited might actually be involved, even if it's not involved in ME/CFS, might be involved in other diseases. And it could affect any part of the body, including the brain. The brain ought to be very sensitive because it needs a lot of pathways. So there are about 80 enzymes that are substrate inhibited. We were looking at the tyrosine pathway because it makes dopamine. And it's a 2-enzyme system as well. And one of those enzymes is substrate inhibited. The other enzyme that's present that could get you out of that is defective in all the patients. So there are multiple systems that we have to explore in this pathway. Our guess is that we really have to set up cultured cells to do this and see if we can actually put the cells into a metabolic trap, and if we can we get them out? And that's sort of be the next wave of experimentation. Meanwhile, we put up all of our data on a server. Most of this has not been published. It's a practice to try to stimulate the community in doing things. So all of our sequencing data is there. It basically means that you can use, anybody in the world can use the data. And that hopefully will help other people that there's very little resources in this disease. And they have to have a good hypothesis, and they also don't have any data, and they can't generate a good hypothesis. So maybe this will allow other people to generate a hypothesis with this data, and they're free to do so. Because this is a really very common, very bad disease that we really need to solve. Thank you.
Thank you so much, Professor Davis.
Thank you.
I know, number one, we're all sort of rooting for the work that you're doing and understand that the answers can't come soon enough for the many people like your son and others who suffer from this. two questions from with the psychiatrist hat on. I mean, one observation, first of all, is that how valuable it would be. I mean, we have so many enigmatic diseases. I mean, even if we just think of psychiatry among the many specialties in medicine, but think about schizophrenia or depression, right? You know, we know so little about the etiology of any of the disorders virtually any of the disorders we treat, and how helpful it would be to have a basic scientist partnering together really on the trail of, you know, thinking so broadly the way you have about possible, you know, possible pathways and causes. That's just an observation about how valuable it would be for partnership of the kind that you've described. In psychiatry, as in with CFS, we have syndromic definitions. So, you know, we sort of define a given disorder like major depressive disorder by certain, you know, symptoms, and syndrome, and schizophrenia same way. And we haven't made a lot of progress in terms of trying to figure out pathophysiology. And one of the ways we've thought about that is that there's probably a lot of heterogeneity. And so we're talking about major depressive disorder as if it's one disorder, but it might be 17 disorders. And so that if we look at 100 people, you know, we're sort of missing the signal. I'm just wondering if that's an issue, or does ME/CFS seem like a more cohesive disorder where it feels like there may be one answer rather than a bunch of different disorders that mimic each other?
Well, I may have distorted that a little bit because I want to make the assumption at the beginning that it is one. It may not be true, but there is a problem, and that is that this disease affects a lot of the metabolites, both raising and lowering 10%, but very significantly, sometimes orders of magnitude. And we also know that in metabolism, you can have mutations and so forth. And generally speaking, heterozygous mutations don't have a phenotype. If you do, and we've done this in yeast with every gene, only a few show a phenotype, and just looking at growth. That's because one gene is sufficient to carry out the function. But we're really dubious that if you start messing with some other parts of the pathway, like the substrates are now very low, probably heterozygous mutations now do have a phenotype, and we've certainly seen in this disease some cases of that. What that will look like is heterogeneous disease, right? And you can even track it back and say, oh, it's because of this gene that's mutated. Yeah, but it's not, you're getting distracted. So I would argue that maybe the number of diseases that you have in ME/CFS is equal to the number of patients. And that isn't helpful. So we have to be careful not to jump to that conclusion, unless it's helpful. Right? And if we find that this classification of the disease is helped by this drug, I don't care that it may just be one disease, but that helps these patients because they have this symptom, and you can diagnose them and you can help them. So I worry about getting too distracted and saying it's a whole bunch of different diseases when we don't know that. And also, one of the arguments that NIH needs to spend more on this disease is because I lump them all together and it's 1%. If we had 10 different diseases, then it would be 0.1% for each one. Oh, then we don't have to spend money on it. Right. So we got to be careful to not split too much because it gives the bureaucracies reason not to support it. And certainly mental illness is a serious problem, and maybe we split it up already too much. And we should lump them all together in what fraction of the population. It's a very high fraction of the population. It's at the moment, it's just a philosophical point of view. I don't know the answer.
Ron, that was a wonderful talk. You know, we see a phenotype. You know, the one IDO is interferon gamma. Can you hear me now?
Yeah, you got to hold real close to you.
Okay. One of my, the IDO mutation, one of them is interferon gamma regulated and the other is not. And we've seen a phenotype with tuberculosis in one and another phenotype in the double. We've just made those mice, but you know, it might be interesting to, as a probe, you know, various, like a TB infection in cells from people with these mutations, it might give a hint. At the same time, I realized the complexity of this. I guess I was wondering too, are you at the point, like, you know, we've sequenced my genome and the genome of my sisters, and we find that I'm homozygous recessive for a spliceosome component and something that's specific in the retina. And, you know, now I find that, you know, many of these RNAs that we think about are really long. And so people, when they're starting to do the packed biosequencing of mRNAs, RNAs find different lengths of RNAs. And so are you moving toward, can you do that sort of analysis on any of these patients?
We've done a fair amount of typical RNA sequencing. It's not enough. It's expensive. And so with this project, it's a matter of when we get a donation, we can do an experiment until we get some sustained funding. And so we then have to prioritize, which is just a guess. And we're constantly guessing this might be a better option to use the money than that. And that's what it's always, and especially if there's some literature that do we have to redo the experiment. It also depends on who we have in the lab. What is their expertise? For example, all this metal analysis was done by a person that was a civil engineering training. So she totally knows how to deal with all these metals and concentrations and so forth. So it's a complex equation as to what we do, but it comes down to having money. And that is a real serious problem for everybody working on this disease. And it's a fantastic thing to have a grant that runs for 5 years. So you know that you can have experiments done for 5 years. In our current state, you know, we don't have that. We have one NIH grant with Mark Davis. So that gives us some funding long term, but it's not a lot. Yes, please.
How accurate are you, let's say, or others at diagnosing disease correctly, just clinically?
Well, I'm not a physician. So
okay, well, others who work with the illness.
Yeah, I can do it, but I'm not legal. So we work with other physicians.
And how good are they?
And I'm very fortunate that in the Bay Area we have a number of really good physicians that work on this disease and are pretty good at diagnosing them. And the two patients that found this cranial instability was one of the physicians that we work with. Three of those physicians now come to our scientific meetings. So we're interacting a lot. We're trying now to set up at Stanford a center of excellence for ME CFS because it's combining the clinic with you know, we look at the patients to confirm that the physician is correct, that they didn't make a mistake. We do get patients in occasionally that are self-diagnosed. Sometimes we will take a blood sample. It's not very expensive if they consent, but it's put into a separate category. We run the analysis, but we don't use the data as mixed in with the other. It's always kept separate as a self-diagnosed patient. Those could easily be something different. And but in the case
you don't have a number like 90% or 80% of the clinicians who say that's the disease, that it's then confirmed.
With the physicians that we're working with, it looks to us like it's 100%.
100%.
The self-diagnosed, no.
Yeah.
They're a mixture.
Thank you.
And also what we do when we have a patient in and the technician who's extremely good says, there's something funny about this patient. It just doesn't strike me. She's not a physician. So what we do is to call up the physician with permission And could you confirm that this patient really has the disease? And we found one of those cases, and he said, well, she kind of meets the criteria as far as I can tell, but just working with her, I don't think she does. And that's that kind of gut feeling that the doctors can have, and I think that should not be ignored. They're not picking a particular symptom, they're just looking at working with the patient overall and saying, there's something funny about this. So we said, okay, she's excluded. And then we ran the nanoneedle out there and she's normal. But we excluded her before we ran it. So that's just an illustration that I think these doctors are actually quite good. And it's a very complex disease, but they use there's multiple criteria, we use them all, because in research you want to be excluding anybody that's doubtful. And in clinical practice, you want to include everybody because you want to try to help them. And sometimes it doesn't matter what the disease name is. You try to treat some of the symptoms and try to guess the best you can to treat them. So there's two sides to this. And with research, we want to be very, very restrictive. That doesn't mean we don't take samples and analyze them and know that they're there. We will continue to do that as well. And we also like having the local patients because we often have them come back to do longitudinal studies. One of our first patients that we started looking at was my graduate student who came down with a disease. And she was sick for well over 5 years. She supported herself because she, in bed, could write smartphone applications and post them for Androids on Google and Apple, and made enough income to survive. And she's now totally over the disease. And I wish we had lots of longitudinal samples on her all along to see what happened. We didn't. But she's clearly over the disease. She's intellectually, as what I can tell, like before. There's no permanent damage that I can detect. And she says, I can run 10 miles and it doesn't affect me. And I said, well, most people can't do that. And so that's encouraging, that this very debilitating disease appears, and she was bedbound, does not appear to cause any permanent damage, which I find a little surprising. But that should be a lot of encouragement to the patients. All the more reason why we need to figure out a cure, that we'll put these patients back to normal. And then one of the, I think, amazing kind of problems is that you can add up the cost to the federal government for this disease. These people don't work in most cases. The severe patients have to have at least one caregiver, sometimes two, because they need around-the-clock care. And often it's a family member or two family members that give up their jobs to do this. And then, of course, there's all the problems of the support and the clinical care and everything. It adds up to about $20 billion per year in the U.S. for this disease, and lost income as well as treatment. And the NIH spends something close to maybe 10 million. And there's 20 billion cost to this disease. I think that's way out of line. And of course, it's all compartmentalized. It's different agencies that are doing the paying and so forth, but it's something that ought to be considered. We would save the government a lot of money if we cured this disease. Yep.
I agree fully. Thank you so much. I just wanted to thank you so much, and Dr. Emmons so much for introducing us to a fascinating area of research on a devastating condition. And we wish the science well. And
your conference is scheduled to end in 2 minutes.
Yeah, let's try to communicate that this disease is out there and it's very common.
Thank you so much.